All right, let's go ahead and get started. So last week you plated the seawater onto the SWC plates, so we should ideally find some colonies. Ideally, we'll find some that are bioluminescent. Remember, we're trying to get a colony that was started by one initial bacterium, which came 2, 4, etc. So that colony represents pure descendants, so they should not have any more than one species in there, okay? So that's our hope. What we're going to be doing is we're looking for bioluminescence, so we'll turn off the lights in the room. If you hold the plate off axis, so you're using your peripheral vision, you have better ability to detect the light bioluminescence that's there, and then look at that, take a Sharpie and mark on the plate. That way when the lights come on, you know which ones are bioluminescent, okay? So what you're going to do is you're going to streak them out. You're going to use a sterile loop, go in there, grab some of those cells and streak out, and we'll show you the streaking technique, okay? The other majority of the lab will focus on enzymes. So you've learned a lot about enzymes in the 1A lecture. Do they change the delta G of reaction? No. Do they change the equilibrium? No, but they do change the kinetics potentially, okay? So this is your chance to actually test enzymes, see how the environment changes activity. We'll test the effects of pH. We'll do what's called the KM experiment, various things like that, all right? So anyway, review with microscopes. You won't be using them this week nor next week, but you will use them again. So always make sure you use that cold illumination. I tried to help a lot of people last week. I was not trying to be a jerk when I came in and said, don't share microscopes. The thing is, you want to use your own microscope. You paid good tuition. You might as well get your money's worth, okay? So it's fine to share slides, but the key thing about the microscope is actually using it, setting cold illumination, finding the specimens. So it's fine to share slides because there's no real requirement to do that repeatedly. But please work with the microscope on your own because you'll learn the skills quicker. So as I mentioned, we should hopefully have a bioluminescent colony. This time what you want to do is there's going to be a variety of colonies on the plate. Variety. Some will be bioluminescent, some will not. Some will be big, some will be brown, some will actually digest the agar that's in there. So what you want to do is look for a bioluminescent one. Again, we're going to turn off the lights. Your eyes will adjust fairly quickly. If you hold it off axis a little bit, you'll see a little bit better. Take a Sharpie, put a mark there on the plate. Some students try using their cell phone for slight illumination. You can try that. For me, it's too much. It bleaches out my retina and I can't really see the bioluminescence. You can try that though. Mark it, then when light come on, you know that's where the colony is, okay? Then you go in there, pick that with your loop, and what you're going to do is do several streaks, each time grabbing a new sterile loop. That's a key aspect. Because what you do is when you go in there and grab that colony, you're going to be grabbing tens of thousands of bacteria. Then when you streak them out, there are potentially tens of thousands in that streak. So when you get a new loop that's sterile, you'll cross this line, pick up a few of those, and spread out the numbers of those bacteria. Again, what we're trying to do is spread them out, and then you take a new loop, cross this line again, streak, and again. So ideally, what will happen is, clearly the density, the number of bacteria along this line should be decreasing. So that when we get to the end of the streaks up here, next time when we grow these up, you should have a colony, and that should have been from a single strain bacterium. Is that clear? So that's the purpose. We'll do that several times. It is really critical to make sure we get that isolated colony. So again, next week, so there's no lab lecture next week. It's on photosynthesis. You'll be doing the streaking again. You do have lab next week, though. You'll need to watch the webcast from last fall. But again, the purpose is to get that nice isolated colony for PCR, which we will talk about in 2 weeks. I did it last time. We're going to go over it again. There are not many times I ask you to do things, but I will ask you to draw out PCR for 3 rounds of replication. And the reason why I ask you to do it is not because it's busy work, but because if you take those 15 to 20 minutes to draw out the 3 rounds of replication, 95% of students understand it right then and there. Okay? Likewise, when I ask you to draw out meiosis, mitosis, please do it. It really will help you learn, okay? So anyway, so what we're going to do is we're going to pick that colony, isolate it, and we're going to do the PCR. So basically, remember, we have a target sequence. In our case, 2 specific genes, the 16S ribosomal RNA gene, another gene called RNA polymerase B. Okay, so RNA polymerase B. So what we're going to do here is keep in mind that for our purposes, we have that large bacterial chromosome, and our target is a very small percentage of that. So the way we target that specific, those 2 loci is we have primer pairs. They come in pairs. One is called forward, one is called reverse. So one will bind like that, the other one will bind like this. This is a primer pair for this genetic locus. If you want to call this one the forward, fine. If you want to call it the reverse one, fine. It doesn't matter as long as you realize they come in pairs, one called forward, one called reverse. Okay? There's no convention about which one's which. Likewise, for the other locus, it will have a different set of primer pairs. But again, the key aspect, they come in pairs. Now, as you know, DNA is double-stranded anti-parallel. So this primer, if it will anneal to this strand, which way is this running? That way, 3' or this way, 3'? So is it running to the right, the 3'? This is the 3' end of the primer. So for it to anneal, it always has to be what? Anti-parallel. So if the primer is like this, that means that template had to be running in that direction. They have to be anti-parallel. So that's why I draw them in 2 different directions. This one will anneal to this strand, and this one will anneal to that strand, following base pairing rules and being complementary and anti-parallel. Okay? Key concepts about those primers. They bind specific locations because of the base pairing rules, and they're anti-parallel. Okay? Now the question is, how do you design these primers? That's really difficult to answer. For now, just pretend we know that these primers are unique for this gene and this gene, and they come in pairs. Any questions about the primer aspect? So 2 pairs of primers. So the way this is work, let's just follow one of these potential targets. Remember, in our case, it's in a big background. But to make it easier to understand, we'll just put it in a small background here. Here's our target. There's there. This primer anneals, anti-parallel, base pairing. Likewise, on the opposite strand, which is complementary in the template, this primer anneals, creates a 3' end. This extends in the extension step. This extends in the extension step. And then we stop it, and we'll separate those. So we denature, which is separation. We do that by heating it up. The hydrogen bonds break. We then lower the temperature. The primers are in the test tube. They're in the background solution. So as the temperature drops, you start to form those base pairs. And since they are very stable because the hydrogen bonds they form, at that temperature, the primers anneal. And then when you raise the temperature and allow extension to occur, the DNA polymerase will extend that using this as a template, and extend that using this one as a template. And then to do the next round, you'll heat it up, which would separate these 2 double-strand molecules into 4 single strands. They serve as templates then. The primers will anneal, and you lower the temperature and extend. And that gets repeated over and over and over again. So this is just trying to show you. Now, there's always confusion about what do I mean by unit and non-unit length. Okay? So I'm going to put that on the board. And then, so what I mean by unit length is if this is my primer here, and this was my target, and this was my other primer here, and my extension, and I'm sorry, let's see if I can use this color. So the white will represent the extension product. The color one represents the primers. If this is exactly it, that's unit length. It means my primers flank in the product. If it's non-unit, that means one or possibly even both of the strands extends past the unit. So this is unit because it's flanked on each end by the primers in terms of the sequence that was made. And this one is not unit. True, it's that size there, but over here, if you notice, it extends past there. So this is non-unit, this is unit. Unit, flank, each end by the primers, non-unit, extends past. Okay? Is that clear? In 2 minutes, there'll be lots of questions about what do I mean by unit and non-unit. So do primers always bind to the 3' end? Primers will be anti-parallel. So what happens here is if I have a sequence like this, double-strand DNA like this, and pretend it extends out and out really big, I could say this is the 5' end and this is the 3' end. There's polarity to the DNA because the phosphodiester backbone, right? So there's polarity. So what will happen is when I separate these strands, let's call this strand 1 and strand 2 so we keep track. Here's strand 1, goes on, and here's strand 2. The way I separate those is by heating them up. So in this case, for a primer to anneal, it has to be anti-parallel. So it will anneal like this because here's 5' to 3', here's 5' to 3', and it will base pair. So that's why it's targeted to a specific strand, because it has to be anti-parallel, and it's why it's targeted to a specific location, because it has to base pair. Likewise for the other one here, and I'm sure I've lost my red dye, unfortunately, red chalk. So this one would anneal, let's say, here, again, 5' to 3', forming my base pair. So what the primer does is it does create a 3' end, and we will see that synthesis, extension, adding the nucleotides, always occurs at the 3' end in this direction, okay? So since it's adding here, it will grow in a 5' to 3' direction. So when we talk about growth, it's growing from a 5' to 3' direction, okay? Did that answer the question? Yes. Okay, so I'm going to show you, last time I showed you the animation, I'll show it to you right now, okay? So I'll illustrate with the animation what unit is and non-unit is, okay? Yes, question? You just take and increase the temperature, and then the DNA strands separate. So in fact, when we do the extension, we do it for a specific length of time, so it keeps extending. But what we do then is we heat up the temperature, which causes the strands to separate. So if the strands were to separate, so if this is going along, and this is a huge background template, if this is going along, but then we take and increase the temperature, the strands separate, and no longer have this as a template to base pair with, okay? The other thing is we may run out of template, which I'll show you in just a second, okay? So there are two reasons for that. So let's just look at the animation. So hopefully the animation will help clear up some of this. We did this last time very quickly. And the animation is on BSpace, by the way. So it's on there as a read only. So you're more than welcome to step through this. Okay, so anyway, we can ignore all this background. We are really, so here's the basic principles. DNA, double-stranded, anti-parallel, stress that over and over again. The polymerase, the thing that makes the DNA extension, requires a primer. It always goes in a 5' to 3' direction. So here's what happens. Always denaturation, separating, annealing, having the primers bind, and extending. And you do that over and over and over, okay? So here's what happens. The template is this length. There's one primer, primer number 1. Would that primer anneal to the upper strand in the template or the bottom strand in the template? Bottom, because it's anti-parallel, and it forms the base pairing rules A with T, T with A, G with C. And a second primer, so this would just show where that primer would anneal. But when we do the reaction, they come in pairs. Now I could call this forward and reverse or this forward reverse, but they come in pairs. And this one now will anneal to the upper strand. And notice, the primers are at the ends of the targets. So when we make the product, they can only be unit length. Only be unit length, okay? So let's just run through that. So we separate, denaturation, just heat it up, anneal, extend at the DNA polymerase. It goes on, and in this case it runs out of template, so it can't keep going, okay? So that's what sets the length there. Now we have two molecules exact unit length because they're flanked by the primers on each end, okay? That's unit length. So you just do this. We go from 1 double strand to 2 double strands, 2 double strands to 4 single strands, to 4 double strands. Again, always unit length. Then we go to 8 single strands. And what we're ending up doing here is always doubling each and every round. Okay? But that's because our target initially was of that unit length. Question back there. So let's do the next thing. So the question was, if you didn't run out of template, would it keep going? The answer is yes. Sort of the answer to the question earlier. So now let's do that scenario, okay? Now let's do what really typically happens, which is where we are looking for a PCR product in the background of a huge target. So here we will see that some of our products are non-unit length, okay? So let's just do that. So here's our target in this big background. And this is not to scale. The background is much, much, much bigger. But to make it easier, we're just doing a subset of it. So now, notice the primers anneal, and they're internal. They're not going to flank the products, and we extend. Okay. Now, this is showing it extending until you end out of the template. But if it was a big, long molecule like this, it would just keep going. But in another minute, we heat it up, and so it would separate. So it would run out of template. Okay. This shows it as though it gets to the end of the template. But in reality, when you have a huge template, just the next step of heating it ends that. Is that clear? Did that answer that question? See that? Excellent. So now we separate that, and just look back to this. Those two are the unit length or non-unit. Excellent job. Non-unit. Thank you. We separate them. 4 single strands. Primers anneal. Again, anti-parallel following base pairing rules. They extend. Are these 4 of unit length? No, again, non-unit because they extend past that. But notice something here. This one will give us unit length in the next round, and this one will give us unit length in the next round. Let's just check that out. We separate those. We have 8 single strands. We bind the primers. Look at this. This one is what? Unit or non-unit? With this one? What are the other 6? So in fact, non-unit sizes always increase by 2 every time. So let's just do some math on the board. It's not real complicated math, but we'll just do it on the board. We start with 1 double strand. After the complete round, we'll go to 2 double strands, to 4, to 8, to 16, to 32, to 64, to 128, to 56, 512, 1,024, which we'll say is about 1,000, which is the same as 10 to the 3. Okay? You do need to know some math like that. Okay? So watch what happens. I said it increases by 2 each time. Non-unit. So this is non-unit of 2, and I'm going to show you a table of this shortly. Non-unit of 2. The total is 2. So how many are unit? 0. Because 2 minus 2 is 0. Again, we always increase by 2 each round in number of non-units. So what will be the number of non-units here? 4. 4 non-unit. There's a total of 4, so the number of unit is? Excellent. Thank you. Now here we go to 8. How many are non-unit? 6. How many are unit? How many are non-unit here? 8. How many are unit? 8. 16 minus 8 is 8. What should the number here be? 10. What's the unit? 22. Notice, we're starting to get a rapid exponential increase in the number of units, but the non-units always increase by 2. Always by 2. So here we're at 12 non, and unit is 52. Here we're at 14 and 114, 16 and 240, 18 and whatever, okay? So you can see that the units are increasing exponentially, and the non-units just go up by 2 each time. Is that clear? Now, if you draw this for yourself, color-code it, it makes a lot more difference than me going over it over and over and over again. But we'll do it again. We separate. Remember, template's huge. Primers anneal internally. We extend. The 2 first ones are bigger than what we wanted, so they're non-unit. We separate, anneal, extend. These 4 are bigger than what we wanted. We separate, anneal, extend. Now we have 8, but 2 are of unit, 6 non-unit. If we separate those 16, the 8 double strand is 16, we're going to have 16 double strand, and of those 8 would be non-unit 8 units. So that's the math behind it, okay? So you can take a look at this. Here's just a table showing that we just did. I did on the board. And the formula is as follows. So this is the number of unit cycles, n is the number of cycles. So 2 to the 10th means you've finished 10 complete cycles. So 2 to the 10th was 1,024, math-wise. So 2 to the nth is the number of total products. The number of non-units is 2 times the number of rounds. So if we had 10 rounds, how many are non-unit? 10 rounds, 2 times 10 is 20. So it's the number of units is the total minus the number of non-units. Okay? So I'll do that on the board here because we did it this way. So the total number is 2 raised to the nth. So if it was 10 cycles, it's 2 to the 10th, which is 1,024. The number of non-units, since it's always increased by 2 each time, is 2 times 10, which is 20 non-unit. And that means the remaining has to be unit, which is 1,004. Because it's n to the 2 minus 2n. Okay? It really will help to draw this out for yourself, okay? So anyway, on the lab exam, I may ask you at the end of round 14, how many are unit, how many are non-unit, okay? So let's just quick do that math, okay? So if I have 2 to the 14th, how do we do that math? Now, I got some emails from students saying, why do we need to know chemistry and math for biology? It's just important, all right? So 2 to the 14th. You can say that's the same as 2 to the 10th times 2 to the 4th. So this is about 1,000. And 2 to the 4th is 2 times 2 times 2 times 2. 4, 8, 16. So that means the total is about 16,000. How many are non-unit? 28. 2 times the number is 28. And then that would mean the number non-unit, unit would be 15,982 or 72 or so, okay? And I'm not going to ask you to do 1,024 each time. Just do 10 to the 3, about 1,000. So after 30 rounds, 2 to the 30, that's the same as 1 times 10 to the 3, times 1 times 10 to the 3, times 1 times 10 to the 3, or 1 times 10 to the 9th. The functions of, exponents, okay? All right, so I know some of you have to wrap your head around the math again, okay? Any questions about that? Yes? So there was no plasmid involved here. This was actually not the plasmid, but the bacterial chromosome, the huge background. So this wasn't the plasmid, this is the bacterial chromosome. We don't need to do that for the PCR reaction, okay? And again, we're not talking about plasmids here at all. This is the background, this is the chromosomal DNA of the bacterium, okay? Bacteria do have plasmids, and we could do PCR on plasmid DNA, but in this case, it's just the background genes within the genome of that bacterium, okay? All right, so there was another question? Was it answered? Yes. So excellent question. So what if you have more than one template? So in fact, the question I asked on the lab exam was, you start with 2 molecules of DNA, and you do 10 rounds, how many should you have? 2 times 1,000, which would be 2,000. Make sense? Because the first time you started with 2, that means your n number has to be multiplied by 2. And in fact, only about 20% of students got that one right, okay? Which kind of surprised me. And I am at fault for that because when I teach this, I always start with 1 because the math is easier. And we do that a lot when we teach. We start with the simplest case, present it. So if you start with 4, and you have 10 rounds, then it's 4 times 1,000. Because that was what we started with, okay? If I started with 2, and I did the 10 rounds, it's 2 times 1,000. So yeah, so if I start with a different number of templates other than 1, I have to take that into account mathematically. So good question, okay? And in fact, if you go through the exam readers, you'll see the variations of this type of question I do. And it's just that very sort of thing, okay? Any questions about this? All right, so let's just end the PCR stuff then. Yes? Right. Okay, so let's just do this. So if we start with 4, double strands. These are double strands. So then I'd go to 8, 16, 32, 64. Tell me when I reach to 10, okay? 256, 512, 1,024, 2,048, 4,096. I think that's it, right? So it's 4 times 1,000, okay? So the question is, do you have more non-unit replications? The math is always the same. So in this case, if we did the number of non-units, since I'm starting with 4, I would have to take that non-units and it'd always be multiplied by 4 then, okay? So because it is the original templates that causes, the original templates are huge, right? They're always in the background. That's why it always increased by 2. With 1, in this case, it always increased by 4 because that's what we start with, okay? All right? But I've never asked that question, but it potentially, okay? All right, so let's get back to lecture. Now, this is an example where sometimes you just have to go back home and do some application and thinking about it, okay? So let's just run through this. All right, so we've discussed that. We did the animations. Remember, what's unit? Flanked by the primers, okay? That's just that table. We've done it on the board. For this experiment, we're going to use micropipetters to add various volumes, so make sure you use the tip. We'll be using the specs again. So remember, you can use the specs to quantitate or identify. In this case, will be the quantification of it. And you should sort of remember that formula.